So hello friends, welcome back to my channel Medicine of 101. Today we'll talk about glycolysis from the aspect of biochemistry. Okay, so let's continue the glycolysis. So before starting, let me tell you guys, glycolysis is what is the universal or the catabolic pathway in terms of the glucose. Okay, so where we'll see the glycolysis that will take place within the cytoplasm of the cell. Okay, so let's write it over here that glycolysis is the catabolic pathway. In terms, you can also universal catabolic pathway where it will take place. It will take place within the cytoplasm of the cell. Okay, so cytoplasm of cell, because see the enzymes that are required to conduct the process of glycolysis, all the enzymes are present within the cytoplasm of the cell. That's why the glycolysis that will take place within the cytoplasm of the cell. Now glycolysis, you know what, first we'll get over here the glucose, and this glucose that will go under the multiple steps, the catabolisms, then it will get converted into what? To molecules of the pyruvic acids in the middle. Okay, so we'll get our 2 molecule of pyruvic acid over here. Okay, now try to understand that in this process we know there will be productions of energy. Now see, in these cases we will get what? ATP. In these cases we will get what? NADH. Now see, to get the energy from the NADH, it is supposed to go within the electron transport chain or electron transport system to get the energy from it. Okay, we'll talk about all these things whenever I'll just discuss in details of the electron transport chain. Now see, so whenever we'll see this pyruvic acid is come over here, under that it will be the what? It will be the initiator molecule for what? Conducting the TCA cycle. Now try to understand, whenever we are getting the pyruvic acid over here, okay, pyruvic acid, after that it will get converted into what? By the integration into the acetyl CoA. So acetyl CoA, then it will go under what? The TCA cycle, or we can say Krebs cycles. Okay. Now see, this process, that means the glycolysis from here to here, it happens where? It occurs within the cytoplasm. And whatever these things that TCA cycles or Krebs cycle, whatever you can say, and the electron transport chain or electron transport system, that will see where? Within the mitochondria. That means the cells that does not contain the mitochondria, where we will not see the what? TCA cycle or the Krebs cycles, where we will only see the glycolysis. That means see, in our body, if we just talk about in terms of the RBC, this RBC, they don't contain what? The mitochondria. So in this case is lack of mitochondria. So mitochondria absent over here. So we will see only here what? The glycolysis. We will not see the TCA cycles or the electron transport chain. Now we will talk about in details about the glycolysis. Okay. Now the first thing, whenever the glycolysis process will be started, whenever we will see there is the presence of glucose within the cell. Now we will talk about how the glucose is coming within the cells for these things. Okay. Now let's talk about the glucose incoming within the cell. Okay. Now see, the glucose that can be found within the cells in the two ways. Within the facilitated transport, and another is active transport. Okay, so let's talk about the facilitated transport over here. And we'll talk about the active transport. Now see, from the term, it's very easy to understand. Whenever we are talking in terms of the facilitated transport, definitely what? There will be no requirement of the energy. So energy that will be not required. Okay. And in terms of, when you are talking in terms of the active transport, here energy utilizations. Here we will see the utilization of energy. Okay, we will see energy required. Energy required. Okay. Now we'll talk about how the glucose is coming from the GI from the GI to the general circulation. From there it will go to the liver. After that, from the liver it will be what? Spreaded in among the what? All what is on within the what? all body tissue or the cells parts. Okay, let's talk about these things. Now I'll talk about first the facilitated transport. In facilitated transport, there should be some transmembrane protein that will help the glucose to go where within the cell. In terms of the active transport, where we will see the utilization of ATP. Now, think about one thing that whenever we are eating or whenever we are taking the carbohydrate-rich meal, for example, after eating, that means that during the feeding state, what do you know that there will be what? We will get here glucose during the feeding state. Now after that we will see from here total circulation, through the total circulation, it will go where? It will go within the liver. Now if I want to draw a little bit of the liver part over here, it's not working, so let me show this one. Now it will go within the liver. Now see, whenever it will come within the liver, then we will see within the hepatocytes it will come over here definitely. And whenever we see there is the abundance of the glucose within the hepatocyte, now very little amount of the glucose that will be thrown out from the liver. For example, from here what? From here through the general circulation, For example, if I see 100% of the glucose is coming over here, most of the glucose, that means near about 70 to 80% of the glucose, that will be taken by the hepatocytes. That will be stored within the hepatocyte. Now see the rest amount of the 20 to 30% of the glucose that will be thrown out within the circulations. Now whenever the glucose will come within the circulation, after that we will, whatever the thing we will see, the glucose that will be sent to the each individual cells. Okay. Now for example, it is the hypothetical cell. Okay. Now see what is the interstitial space. Interstitial space. Now from here, the glucose that will go within the interstitial space. Now it is the cell. Now within the cell, there should be, in terms of the facilitated transport, there should be some type of transporter. That means transmembrane, transporter over here. We will get transmembrane protein. Now within the nucleus, we know each cells there will be the genetic material. From here we will get one type of the mRNA. That's the normal thing I already discussed. From here we will get one type of the protein. Now this protein will come over here, just like this, and this protein will help to take the glucose molecule from here to here in this region, from outside of the cells. That means from the interstitial space to the cell. Okay. Now see, as we know, the concentrations of the glucose outside of the cell. Now right now during the feeding state it will be more. Now within the cell that will be less. Okay. Now what type of the transporter is this? This is known as what? Blood transporter. That means glucose transporter. We just name it as blood transporters. Okay, GLUT. Now we can classify the GLUT in the different ways. There are many types of the GLUT, around the 14 to 15 types of the GLUT, but you're not supposed to know all of them. So what you are supposed to know, I'm writing all of them over here. Okay. Now in terms of these things, we'll get the GLUT 1, we'll get GLUT 2, we'll get GLUT 3, we'll get GLUT 4, GLUT 5 and GLUT 7. Okay. Now see, near about in all cells we will get the GLUT 1, GLUT 3. So GLUT 1 plus GLUT 3, that is present in almost all cells. Okay. But the concentration of the GLUT 1 and GLUT 3, that will be more where? Within the RBC, and that will be more within the RBC and central nervous systems. That means neurons. I am writing it over here. So we will get where? Within the RBC and within the central nervous system. Okay, that's like this. Now what about the GLUT 4? This GLUT 4 we will see where? within the muscles, for example the skeletal muscles, and within the adipose tissue. That is important thing that you need to know in terms of the GLUT4. Now basically we will see whenever we know what within the adipose tissue or within the muscles there is storage of the glucose. Now this type of the GLUT4 that will act under the influence of insulin hormone. How? Let me show it over here. For example, it's the for example during the feeding state, if I just say just like this, is the cells, for example the muscular cells or the adipose tissue cells over here. Okay, now within the cells you will see there is one kind of the receptors over here. Now these receptors is having a within the receptors there is a presence of one type of portions where insulin can bind. So during the feeding state we know that insulin will come over and it will bind over here, and whenever it will come over in these regions and it will get bind with these domain regions. Now it will give the many signals within the cells, intercellular signals. One of the signal that will go where within the vesicles. Now there will be certain types of the vesicle, presence of the vesicles, where we will see the presence of GLUT4. So the GLUT4. Okay. Now whenever the insulin will come over and it will give the stimulus within the cells, then we will see this thing that will come from head to head, and we will see there will be the fusion of GLUT4. Now this GLUT4 that will get fused within the membrane, and they will take the glucose just like this. The glucose will be taken by the what? Muscular cells or the adipocyte tissue cells. Okay. Now see one thing about the GLUT and GLUT3, they are having the maximum affinity for the glucose. The GLUT1 and GLUT3, they are having the maximum affinity for glucose. Think about this thing, that we know that we want our nerves, or we can say in terms of the central nervous systems or the brain, to work always continue. Okay, that's why this the GLUT1 and GLUT3 that will be more, the concentrations of the GLUT1 and GLUT3 that will be more within the central nervous systems or within the brain. For example, when you will see the glucose that is less, that means in the hypoglycemic conditions, yet we will see this GLUT1 and GLUT3 having the capacity to get bind with the glucose and to supply the glucose within this region, that means within the brain. That's the main important thing over here. Okay, and what about GLUT5? The GLUT5 that is present where, that is for basically for the fructose transport. So the GLUT5 for the fructose transport, and where we will see the fructose in terms of the sperm formations, that means within the testis. So within the testicular cells we will see this type of the transport, that means the GLUT5 that will be present within testis. And the GLUT7, we will see the presence of the GLUT7 that will be within the endoplasmic reticulum. Endoplasmic reticulum. I will talk about these things in details later. Okay. For now, just know these things. And what about GLUT2? Yes, this thing is important. Now, where will see the GLUT2? Okay. Actually, think about this thing, that GLUT2, for example, is the cell. Okay. Now the GLUT2, they are bidirectional. That means within the cells the glucose can come outside from the cells to, within the cells to inside of the cells to outside of the cell, and from the outside of the cells the glucose can get within the cells. Okay, this type of thing we will see. Now this type of the GLUT2 transporter we will see, basically we can see in terms of the pancreatic beta cells. So we will see in terms of pancreatic beta cells. 3. Whenever the glucose will come, why it is bidirectional, whenever they in the pancreatic beta cells, then you can understand what about the environment of the, like within the circulations, whether it is hyperglycemic, whether it is hypoglycemic. Whenever the glucose will come, at that time the pancreatic beta cells that will be activated, and that will secrete what? That will secrete the insulin hormone, and the insulin will go over here and it will do its function. That will control the blood sugar level. Okay, that's the main thing. And that type of the thing we'll see. There are the pancreatic. We can also see within the kidneys. So this type of the thing we can also see within the kidney. The GLUT2 transmitters. Now we'll talk about these were all about what the facilitated transport where by the transmembrane protein, or you can say the GLUT transporter, where we will see the glucose that will be taken from the interstitial space to the cell. Okay, that's the thing. Now I'll talk about the active transport. So these are all about the what? These were all about facilitated transport. Now I'll talk about the active transport. Now in terms of the active transport, let me say you over here. Now see, when you are talking in terms of the active transport, where we see the active transport? For this thing, let me draw a nephron, because basically we will see within the PCT, proximal convoluted tubular epithelial cells. We will get it over here. For these things, I need to draw a little bit of the PCT part of the nephron. Okay, the thing like this, and we know that from the circulations, from the general circulations, whenever the blood that will come over here, so along with the blood there will be what? The glucose, the different types of the ions, for example sodium, bicarbonate, this thing will be also here. Now see, after that what? After the filtrations, we will see the glucose that will come over here, the sodium that will come over here, and the different types of the ions, they will also come over here. Now if you see the epithelial cells of the proximal convoluted tubule over here, for example this one, now is the luminal site, is the basolateral site, and whatever the function in terms of the basolateral sites, the functions for the basolateral sites, we know there is the presence of what? There is the presence of sodium potassium ATPase. Sodium potassium ATPase. Now whenever we are getting the sodium potassium ATPase pump over here, see the sodium, the 3 molecules of the sodium that will be thrown out, and the potassium, 2 molecules of the potassium that will be taken in. In the same way, if we just say over here, the 3 molecules of the sodium that will be thrown out and the 2 molecules of the potassium will be taken in the same way, we will see over here. Okay. Now see, if we see this type of thing, then there is what? There will be the deficiency of the sodium within this epithelial cells, and whenever there will be the deficiency of the what? The sodium within the epithelial cells in terms of the proximal convoluted tubule, at the time we will see within the luminal side there is presence of one type of the transporter. Okay. There is presence of the transporter which is having 2 domain. Okay, which is having 2 domain. Within one domain, we will see that sodium can bind. Okay, now normally just think out one thing. Whenever the sodium is binding over here, as we know that the sodium is supposed to go from the higher concentration to the lower concentration. That means, as far as the concentration gradient is concerned, there should be the facilitated transport for what? The sodium, because see, the sodium that is in more in amount, the more in concentrations outside the cells, and the concentration of the sodium within the cell that is less. So the sodium that is supposed to go just like this, but the pump or the the transporter will not in this way, because if the sodium is supposed to go from the outside, that means from these portions, from the lumen to the cell, along with that there should be also binding of the glucose. Now whenever the glucose is binding, after that it will just flip it. Whenever along with the sodium the glucose molecule will also bind, after that what? This protein that will flip in. As a result, we will see that sodium will come also here, and along with that glucose will also come over here. Now for this thing we need what? The energy. How the energy will come? See, whenever the sodium is going out, at the time what? The utilizations of the ATP. So ATP using over here. Now we will consider, for this thing we have to know 2 more things: the primary active transport and the secondary active transport. Primary active transport and secondary active transport. Now see, what is the difference? Now the primary active transport means in the transport or in the protein directly the ATP is getting utilized. So whenever we will see the ATP is directly getting utilized where in terms of the sodium potassium ATPase, so we can say this type of the transport that is known as active transport. Now it will be known as the secondary active transport. Why? Let me show it over here. Whenever the sodium is going from what? From within the cells to the out of the cells, that means whenever the sodium is going from inside of the cells to the outside of the cell, as a result we are seeing the deficiency of the sodium. And whenever the sodium deficiency, see, there will be what? concentration gradient, and the energy whatever it is using over here, the little bit of amount of the energy that will be utilized here. So by using this energy, by using the energy where it will utilize where in terms of the sodium potassium activases, the energy in terms of the secondary active transport it will be utilized over. Along with that we will see the sodium and the glucose that will be transported within the cells. In the same way we will see this, there is a bind for the sodium, a binding site, sorry, there is a binding site for the sodium, and there is also binding site for example for the amino acids. So by using the secondary active transport mechanism, by you see by by the mechanism of the secondary active transport, this sodium will be taken within the cell along with the amino acid. Only the glucose or only the amino acid that will be not taken. That along with that the sodium is supposed to go from the lumen to the within the cell. That's the thing. And for these things we have to use the secondary active transport. Active transport that will be utilized where in terms of the sodium potassium ATPase. That you have to understand over here. Okay. Just like this, we already got the glucose within the cells. Now the glycolysis process that will be studied. Okay. Now the process of the glycolysis that will be studied over here. So let me tell you first. I have already said in the first part that the first example, when the glucose is coming within the circulations, and from the circulation the glucose can be transported to the cells by two mechanisms. One mechanism is facilitated transport, another is active transport. In terms of the facilitated transport, I have already said over here that different types of the GLUT transporter, GLUT1, GLUT2, GLUT3, GLUT4, GLUT5 and GLUT7. And in terms of the active transport, where we will see the secondary active transport by which the sodium that will take the glucose molecule along with it and it will be taken within the cells. After that the glucose will be within the cells for the glycolysis process. Okay, just like this we will get the glucose molecule within the cells. So this was the first part of the glycolysis. In the next part I will start actual the glycolysis, what happens within the cells after taking the glucose within the cell. Okay, so just keep watching for the next part. Okay, thanks for watching.